the anatomic pathology supervisor at the Willistown Hospital. So I'm going to talk just briefly about any careers that you might choose in the clinical laboratory, and then get into more specific into pathology and what we do in pathology. And then we're going to do some hands-on staining, which is a big component of what we do in pathology. I'm going to forward this to Ms. Shade, and then if you guys are interested, just because you don't have a projector. Okay, so the lab is a pretty relatively unknown department in a hospital, but really, really important. About 60 to 70% of all medical decisions are based on the laboratory diagnosis. So nothing is made solely on the histology or clinical laboratory diagnosis, but in the grand scheme of things, putting it all together is really helpful. So there's a lot of different fields to go into if you're interested in working in the clinical lab. There's medical technologists, there's cytotechnology, histotechnology, and then pathologist is the doctor who oversees the laboratory. So medical technologists, they work in the clinical side of the laboratory. They're the people who are dealing with bloods and body fluids. So you go to the doctor and they say, we need to draw some blood. And then you magically get a result like a day or two later. So what's happening in the lab during that time period? So there's people, medical technologists, who are working and they're analyzing your blood and all the components in your blood to give a good example and overview of what's going on so the doctor can treat you if something is wrong. Example of that, a patient comes in to the emergency room thinking they have a heart attack. They're going to draw some blood. They're going to run a test called troponin, and what that does is it measures the cardiac enzymes in your blood. So we'll be able to tell you if you are or are not having a heart attack at that moment. So there's two different levels of medical technology. There's a 4-year degree and a 2-year degree. Medical technologist is a 4-year degree. Medical laboratory technician is a 2-year degree. So they do a variety of things. They do microscopic exams and analysis of blood, body fluids, and cells. They're going to identify bacteria, parasites, and other organisms. They're going to do analysis of body fluids for chemical analytes such as glucose or cholesterol. They're going to cross-match blood for transfusions. So anytime you need to get somebody else's blood, we want to make sure that it's compatible with your blood and your antibodies so that you're not going to have a reaction. And we're going to perform differential cell counts looking for any abnormal cells in your blood. So then moving on to the anatomic pathology side of the laboratory. Again, there's 2-year and 4-year degree professions. There's histotechnologist or histotechnician, which is a 2-year degree, and then a histotechnologist, which is a 4-year degree. There are 2 schools in the country that offer a master's degree in histotechnology, the Drexel University and the University of Tennessee are the 2. So what we do in pathology is we prepare very thin sections of tissue and we put it on a glass slide for a doctor to look at under a microscope. There's 5 basic steps of pathology. The first one is grossing. So what we do is we do a macroscopic exam. So an organ is removed from a patient. It's going to come down to us, and what we're going to do is called a gross description. We're going to weigh it, we're going to measure it, we're going to describe what it looks like, we're looking for certain cell structures. If it's large, we're going to cut it up into smaller pieces. And then it's going to go on to a step called fixation. And what that does is it stops the tissue from breaking down. We want everything to remain as it was in the patient's body, so it's as close to that time period as possible. It's going to go through a step called processing, which what we do is we use heat and vacuum and chemicals, and we're going to remove all of it. It takes anywhere from 1 hour to 12 hours, depending on the size and consistency of the tissue. It's ready for embedding, and what embedding is we're going to create a mold of the same wax with the tissue inside. That's going to form the base of how we're going to make the slide. Microtomy is the next step. That's where we're actually going to make the slides. So we have a machine, it's called a microtome. It's essentially like a glorified deli slicer. Works the same exact concept. A layer of cells is approximately 5 microns thick, so we want to cut all of our wax sections at 5 microns, so we're only looking at one layer of cells. After that, we need to apply dyes to the cells so that we can see what's going on inside of them. If we don't, they're transparent, we can't see what's going on inside. We want to see the nuclei, we want to see the cytoplasm, we want to see the cell membranes. So everything is going to get a routine stain. It's called a hematoxylin and eosin stain. The hematoxylin is going to stain the nuclei purple. It's going to stain the cytoplasm and the muscle pink to red. There's some other careers in anatomic pathology. There's cytotechnology. They're the counterpart of anatomic pathology, but they're looking for malignant cells in body fluids. There's pathology assistant, which is basically the person who's in between the histotechnologist and the pathologist. They're the people who are going to be doing a lot of the gross descriptions of the tissue. They're also going to be doing what are called frozen sections. So we can do that whole really complicated 2-day process in about 20 minutes while the patient's still under anesthesia. The pathologist assistant is the one who's going to be doing that. And then the pathologist is the doctor who's ultimately going to be looking at all the work that the technologists have done. They're the ones who are going to be responsible for making the diagnosis. So one note I want to make about laboratory sciences. The laboratory has been predominantly populated by the older generation, which are retiring. So the retirement rates are raising every year. The vacancy rates are incredibly high. So the anticipated retirement rates over the next 5 years for the lab range anywhere from 19 to 25%. So the vacancy rates are growing, which pretty much means in about 2 to 4 years, anybody can have the pick of any job they want. It's something that is just going to continue to need and grow. So it's a really good career to get into. Okay. So just a safety note, histology, anatomic pathology is actually in the entire hospital the most dangerous department in the whole hospital. It's a blend of biology and chemistry. We deal with a lot of chemicals. We have a lot of mechanical hazards. We deal with a lot of really sharp objects. We use scalpel blades, we have bone saws. Microtome blades are designed to cut through bone as well. So we have to be really, really careful and really just pay attention to our safety at all times. I kind of went over all of those steps. Okay, so today what we're going to do is a gram stain. So a gram stain is a very basic stain. It's used to identify bacteria in tissue. Now it can be done in microbiology or it can be done in histology depending on what kind of tissue or cultures are being submitted to the lab. We're going to use this to demonstrate gram-positive and gram-negative bacteria. So we're going to use a chemical called crystal violet oxalate. And what that is going to do is it's going to bind to the gram-positive bacteria cell walls. So gram-positive bacteria have a phospholipid layer around the outside of their cell walls. And that's going to hold onto that crystal violet oxalate. As we differentiate the cells that don't have that phospholipid layer, that crystal violet is going to disappear. We're going to come in with a neutral red, and that's going to bind to these gram-negative. So we're going to use this stain. Normal bacteria is found everywhere in the body. It's on your body, it's in your body. We need it to survive. But the gram stain is going to pick up infectious bacteria. And it's going to be used, the stain itself is only going to take us about 5 minutes to do. So we want to do this so that a patient can get empirical antibiotics before a culture is given. Culture is going to take about 24 to 48 hours to grow, which might be too long for a patient to wait for antibiotics. So we can do this, patient can get started on antibiotics, and then it can be altered once the culture is back. So everybody wants to kind of come over here. We can get started. Does everybody have a pencil? No, have pencils?
Yeah.
Or just a couple
Don't have a
pencils.
pencil. Raise your hand.
Probably only need one. I just want them to write their initials on this slide.
I'll grab a few.
Alright, so we'll kind of spread over a couple of benches. Just have to group together, probably in groups of 2. So if you guys want to come up, get some gloves, and group up, and then I'll bring you guys some slides. Okay, so when we cut the slides, like I said, they're cut in wax. But before we can put any aqueous dye on it, because water and wax don't mix, we want to melt all the wax out. And then we can use into a chemical called dyeing. That's going to get rid of any extra wax, any melted wax. And then we're going to rehydrate it up to water. So that step has already been done for you. So now the wax has been melted, and this is water that's on it now. So if you want to go ahead and just sort of set it a little evenly on there, the clear part facing this way. There you go. Okay, you can get your slide there too. We'll do it together. So the first step is to apply the crystal violet. Just want to set your hands first. So if you guys want to get some crystal violet, what you're going to do is just suck up some crystal violet and put it on the clear part of the slide. And what that's doing is, you can put on somewhere you want to cover up pretty much that whole part. I'm not sure exactly where the tissue is. That's good. So what that's doing is it's binding to that phospholipid. You could throw them in the trash then when you're done. So that's going to bind to the phospholipid layer of the bacterial cell walls. Alright, so now if you want to take your slide and just rinse off the purple under the water. And you notice the water is not on really high. We want to be careful because if we put it on really high, or if it's really hot, the tissue will slide right off. And so you can just set it back down.
I usually memorize these here.
Alright, so the next step is we're going to put some iodine on there. So, we're going to take some iodine. You're just going to do the same thing like you did with the purple. Just put it on the clear part of the slide. Just enough to cover the clear part of the slide. You can see where the tissue is now, those little purple dots. But what that's doing is it's helping the purple to stay in that phospholipid layer. So the next step that we do, which is the differentiation step, which is going to wash all of that purple out, it's going to protect that purple from leaching out of the gram-positive bacteria. Alright, so you guys can go ahead and rinse those off. move on to that. So the next step is differentiation. It's going to be really quick, so I'm going to help you out with this stuff. So if you just want to hold it over there, we're going to use acetone to do the differentiation. So I'm just going to drop it on, and then you're going to rinse it right off. Yep, so you can go ahead and set it down. So you can see that it's purple. All that purple is going to leach out. Alright, so then the final step is to stain the other bacteria. So this is called a this has red, which is going to stain the rest of the bacteria, and then it's got green, which is going to stain all the other tissues that doesn't have bacteria. So if you want to go ahead and drop your slides in there.
Yeah.
Nope, you can just drop it right in. It's going to sit in there for about 2 minutes. So normally after we complete our last step of the stain, we're going to want to do that dehydration. We're going to dehydrate the slides. But this is an alcohol-based solution. So if we run it through alcohol, it's going to leach out all the stain. And we're going to end up with the tissue again. Not do anything. So once this is done here, we're just going to rinse off the dye with water, and we're going to let them air dry. And then we'll take a look at them under the microscope. Guys, have any questions about staining? This is a pretty simple stain. A lot of the stains that we do in pathology take a lot longer. We have some stains that take 1 or 2 days to do, or that have to be done at heat or have to be done in microwave. So we can stain for pretty much a lot of different types of bacteria, fungus, different types of connective tissue. You can look for collagen versus muscle. And they're used for IDing different types of diseases, different types of cancer. We can see if like tumors invading, if it's breaking through certain fibers. So we do a lot of different kind of staining. So this is probably one of the most manual parts of the laboratory that's left. A lot of it's been automated over the years. Okay, so if you guys want to take your slides out, use that to grab them. So you're just going to rinse them off with water, and you can lay them on this paper towel when you're done. Yep, rinse off with water. And you can lay it down. You can get it? Oh, there you go. You can lay it here on this paper towel. Yep. All right, you guys are all done. You've done a gram stain.